How does concentration, temperature, and pH affect an enzyme? Catalase is a very common enzyme that acts as an antioxidant. It's present in most living tissues and plays a major role in breaking down hydrogen peroxide into water and oxygen gas. You see, the problem with hydrogen peroxide is that it can cause damage to many organelles within a cell. And peroxide is produced by many of the metabolic reactions within a person's body. And so catalase is necessary to maintain a cell's proper health. And you see, the effectiveness of catalase can be observed by measuring the amount of products a reaction using catalase can create. It's a good representation for enzymatic activity due to its abundance, its importance in biological function, and how much we know about it. In this experiment, we measured how concentration, temperature, and pH level may have affected the catalase and its ability to perform its proper functions. We predicted that an increase in concentration would increase its effectiveness, that both pH and temperature will decrease its effectiveness the further away it is from 7 pH and 37 degrees Celsius, respectively.
So for our methods, we decided to investigate the effects of enzyme concentration, temperature, and pH levels on a catalyst level. We varied each factor across multiple trials and measured the resulting enzyme activity. So catalyst solutions were prepared, at different concentration levels between 5 to 20% in order to observe how different the amounts of enzymes influence the actual reaction rate when combined with a constant volume of hydrogen peroxide. So for the temperature, we had different reactions as well between 4 degrees Celsius and 50 degrees Celsius to determine, the optimal temperature for each activity. So our hydrogen peroxide our catalyst and hydrogen peroxide were equilibrated to each temperature before mixing. And the volume of bubbles produced was recorded, so bubble volume, for each condition. This was used to assess the effect of our pH levels, depending on 3, 5, 7, 9, 11, using buffer solutions. Each reaction was conducted at the designated level, and bubble volume was also measured as an indicator of the enzyme activity. This approach allowed us to examine how changing enzyme concentration, temperature, and pH affects catalytic efficiency by measuring the reaction rate through bubble formation.
The graphs illustrate how catalyst concentration, temperature, and pH affected enzyme activity, measured by bubble volume. Higher enzyme concentration speeds up the reaction as more enzyme molecules interact with the substrate, hydrogen peroxide. However, beyond a certain concentration, this effect may level off if all substrate molecules are bound. Temperature also affected the reaction rate, peaking around 37 degrees Celsius. Lower temperatures slowed the reaction, while higher temperatures, especially around 50 degrees Celsius, led to a decrease in bubble volume due to enzyme denaturation. Excessive heat disrupts the catalase structure, diminishing its effectiveness. Enzyme activity varied across different pH levels. The highest bubble volume occurred around pH 7, which is close to neutral. At more acidic pH 3 and 5, and more basic pH 9 and 11 conditions, bubble volume was reduced. Extreme pH levels likely altered the enzyme structure or charge properties, impairing its ability to bind effectively with the substrate. This indicates that catalase has an optimal pH range with maximum activity near neutral. Beyond this range, its structure becomes less suited for catalysis. The experiment demonstrates that catalase activity is influenced by concentration, temperature, and pH. Enzymes function best within specific ranges, and conditions outside these ranges can impair their activity. This information underscores the importance of maintaining optimal conditions for efficient enzyme function.
In conclusion, this experiment has demonstrated that mixing various solutions produced distinct amounts of bubble volume and pH level because of the properties of each solution. The variation of this bubble volume may have been caused by factors such as the surface tension of the water, its viscosity, and its chemical reactivity, Our research suggested that different solutions interact uniquely with water, which would change the number of bubbles created by a catalase reaction. Let's point out the elephant in the room. These experiments were performed in a digital simulation. These were not actually performed in a real laboratory, and so we are assuming that the results and the data that we gathered were accurate to the results that would have been gathered in a real-life, in-person laboratory. We also assumed that pH, temperature, and concentrations were the only factors that could actually affect an enzyme's ability to perform its functions. If there were confounding variables not known to us, then they were not known.